welcome back everybody to the seminar series of the Anne S. Bowers Women's Brain Health Initiative. Today we are delighted to have a talk by Professor Poldrack. Russell Poldrack is the Albert Ray Lang Professor and Department Chair in the Department of Psychology at Stanford University. He's the Director of the Stanford Center for Open and Reproducible Science and the Associate Director of Stanford Data Science. He's also the Director of the Data Coordinating Center for the Bowers Women's Brain Health Initiative. His research uses neuroimaging, behavior, and computational modeling to understand the brain systems underlying decision-making and executive function. His lab is also engaged in the development of neuroinformatics tools to help improve the reproducibility and transparency of neuroscience, including the Open Neuro and NeuroVault data sharing project and the Cognitive Atlas ontology. Russ, thank you very much for joining us. The floor is yours.
All right. Well, thanks very much for the invitation. Happy to be here and happy to be part of this amazing project. Yeah, so I'm going to talk about how, the work that we've been doing over about the last decade to develop open science tools for neuroscience in general can be leveraged in particular for women's brain health and the Ann Bowers Women's Brain Health Initiative. And I don't think it's a secret to anybody here that women's health issues in general are underfunded by comparison. So here's just one example, a nice analysis that's from Smith in Nature a couple years ago, showing if you sort diseases that differ in their prevalence in males versus females by their burden, you see that there's a lot of them up top. The size is related to the burden. There's a lot up top that are kind of specific to or sort of more heavily dominant females. If you look at the funding, you see that many of those end up being towards the lower end of the funding distribution, such that women's health issues in general are underfunded in terms of NIH research. And I'm sure you're all familiar with Emily's recent work, this piece in Nature a couple of years ago, which highlighted the fact that this is true for neuroscience as well. And I'll come back and kind of dig into this particular point in a little bit more detail later in the talk, but just highlighting the fact that that women's brain health has been severely neglected, and that the Ann Bowers WBHI is kind of aiming to change that. So first, just looking at PubMed to get a feel for what this looks like in a more granular way. If you search for fMRI, you have to put it in quotes to make it specific, but you get about almost 63,000 papers. And if you look for papers that are about kind of, relevant neuroscience topics that, you might think are interesting and kind of niche, but not super niche, you find things like, bilingualism, 378 papers, music, 842 papers, mental imagery, 466 papers. Now, those are all perfectly good things to study, but you would hope that issues like menopause and menstrual cycle and contraceptive use would be at least as well studied as bilingualism and mental imagery. Unfortunately, you'd be wrong. The number of papers that hit for these search terms are far below any of those other topics. So I think this is a really striking, example of the more general point of kind of the neglect of these topics in the literature. So as if you've been to any of the talks here previously, you've heard about the goals of the WBHI, which are really to kind of balance the scientific scales and generate the kind of data that we need to really understand these particular questions. And so I'm going to focus particularly today on the first aim, the big data side, where the goal is really to get data from lots of brains, across kind of a very broad population, and get the data that we need to ask questions about things like, what's the relationship between contraceptive use and brain function and structure? What's the relation with menopause? What's the relation with menstrual cycle? To understand, kind of cross-sectionally those relationships in the population. Now, in addition, we want to dig deep, right? And I'll talk a little bit about how we're starting to do that, as we try to collect big data by trying to collect many more measures, both behavioral measures and physiological measures. And then we have plans to go even deeper. I won't say anything today about the precision imaging part, which is the kind of longitudinal, dense imaging of individuals across many time points to understand how things change over time. And I think, the recent work on mapping the brain across pregnancy was a really nice example of this particular type of approach. But again, I'm going to focus primarily today on the kind of the big data side. I'll have a little bit to say about deep phenotyping, but we really have this kind of broad set of goals in mind. So why big data? Why is this necessary? I'm going to talk about sort of two reasons why we need big data, right? One is to make the answers that we get from all of our science more reproducible, and then in particular for empowering the kinds of AI models that Nina and Amy and others within the WHI want to empower. So let's first talk about reproducibility. And many of you are certainly aware of the fact that over the last decade and a half, there's come to be what people call a reproducibility crisis. I would if you don't know about this, I'd recommend Richard Harris's book, which kind of lays out a lot of the evidence, particularly in the biomedical domain. But it really spans across lots of different areas of science. This is a poll from Nature a number of years ago, where they asked people in many different areas of science, how have you failed to reproduce an experiment of either someone else's or your own? And most that in all of these domains, the majority of scientists have failed to reproduce someone else's result at least once. Clearly, that showing that that's a big problem. I think it became most prominent in psychology early on, but it's become clear that it really is not kind of a specific to psychology. Now, I'm going to talk about this specifically in the context of neuroimaging, by showing like one of the, I think, striking examples of irreproducibility, and that has to do with neuroimaging of depression. So there's been a lot of studies that have looked at the neural correlates of a diagnosis of depression, and there have been meta-analyses that try to combine across these. And so Mueller et al. In a paper a number of years ago in JAMA Psychiatry looked back at a number of these meta-analyses, all of which had claimed to show that there were consistent differences between the brains of people with a diagnosis of depression and those without one. It turned out, so Mueller's group is probably the world's best group at neuroimaging meta-analysis, and they went in and identified a number of problems with the previous meta-analyses. When they put all these data together and fixed those problems, they found no convergent results, no convergent differences between people with depression and people without depression across 99 studies. So 99 papers all claim to find a difference. Put them together, big nothing burger. That seems like a big problem. And we've come to think that a major cause of this problem has to do with statistical power. So we've known, going back to Kate Button's paper back in 2013, that neuroscience studies are badly underpowered in general. So remember, the statistical power is the likelihood of finding a statistically significant effect, assuming that there is a true effect. So what this is saying is that the majority, many neuroscience studies, and this is combining across animal studies and human structural imaging studies, basically had so little, like even if there is an effect, they're only going to find it 20% of the time. Those are studies that a review board would refer to as a futile study. It's not worth putting subjects at risk or killing animals in order to do a study where 4 times out of 5, even if there is an effect, you're not going to find it. Marcus Munifo and colleagues have subsequently done a much broader analysis of statistical power and showed that it's really the case across lots of different biomedical research. The most studies are badly underpowered. Now, you might say, well, that's fine. You won't find most of the things, but when you find things, you can believe them if you do the right statistical analyses. But it turns out that there's another way to look at this that John Ioannidis first pointed out a number of years ago, and that Button et al. Kind of highlight in their paper, which is that what we usually control in our statistics is the likelihood of any false positives across all of our studies. But what we really care about is what we might call the positive predictive value, the likelihood that when we find a positive result, it's true or not. And that depends on both the type 1 and type 2 error rates, and also the likelihood that the study will find that the test that you're doing has a true effect. And so that what Ioannidis and Button et al. Have pointed out is that when power is low, most positive results are going to be false positives, right? And a thought experiment to kind of demonstrate is this: imagine you're a physicist, you've got some detector that's trying to find, something in space, but unbeknownst to you, your detector is broken and it's just giving you Gaussian noise. So you can do 100 experiments. 5% of those, if your alpha is 0.05, 5% of those are going to give you a statistically significant result, none of which are true, right? So your positive predictive value is zero because your power is zero. As your power goes up, the percentage of your results that are true results is going to go up as well. So this drove Tagliarconi to say, a while ago, very presciently, consistently running studies that are closer to 0% power than 80% power is a sure way to ensure a perpetual state of mixed findings and replication failures. And I think that there's a really instructive kind of case study in science that started around the year 2000 when it became possible to kind of reasonably cheaply genotype people for particular genetic variants. And we started to see papers showing associations between particular genetic variants and particular phenotypes. And this is a meta-analysis of studies looking at the relationship between this particular polymorphism in the brain-derived neurotrophic factor gene, which is thought to be related to the hippocampus. We know that it's expressed there, and the volume of the hippocampus. The plot here shows over time the effect sizes of the reported studies, and then the size of the bubble relates to the sample size. So what you see basically is early on, all the results were positive. The smallest samples gave you the biggest positive results. Over time, as sample sizes get bigger, the effect sizes get smaller. And we actually know that now from very large samples that these effects just don't exist. And in fact, for almost every candidate gene study, candidate gene association that was studied, we now know that none of them are significant, with maybe one or two exceptions. Certainly BDNF, now that we have studies with tens of thousands of people, there's no association. There are other genes that nobody had thought of, other variants that have associations, not BDNF and not any of these other candidates. So how well powered I'm going to focus on fMRI studies because that's the area that I work in. How well powered are fMRI studies? Well, we've been tracking sample sizes over time. We first published something on this back in 2017. This is an updated plot from this weekend. So the median study going up through 2024, the median fMRI study in 2024 had a sample size of 36 subjects per group. You can see that's been going up. This is a log scale, so it's actually going up, a little bit better than this linear plot would suggest. So we can ask making some assumptions, we can ask what effect size would that sample size give you the power to find at p less than 0.05 for like a single activation? And that's about 0.67. So that's kind of a medium to large Cohen's d. The question is that a plausible effect size, right? We need our samples to be powered to find plausible effect sizes, right? If I told you I'm going to go do a study that has power only has power to find differences in height between a 4-year-old and an adult, right? I don't even need statistics to find that, right? That's you can do that with 4 subjects probably. That's not a plausible effect for most studies that are interested in phenotypes. So we did an analysis. The problem is you can't look at published effect sizes because they're all based on doing lots of statistics and taking the winners. So they suffer from what we call the winner's curse. They're inflated. So you have to use data where you have some kind of a priori guess about where the activity happens and then quantify that. So we use the Human Connectome Project data. Fortunately, that study had given us prior ROIs. And then we could also use Neurosynth to find, the areas that we think are really likely to show activity for things like a motor task. And then we can ask, what are the effect sizes? So first we can look at the effect size within subject. That's what you see on the left, which is how much does the bold signal go up? We know that in the biggest realistic bold effect sizes you ever see are on the order of 4 to 5%. And that's usually in primary sensory cortices or primary motor cortex. And in fact, we see in the supplementing motor cortex, a percent signal change of about 4%. So that's about the strongest effect you would ever expect to see. What is the Cohen's d, right? Because this is looking within subjects. When we do statistics across subjects, we have to look at the variability between people. And that's how you compute that Cohen's d. It turns out that it's right around 0.67. And most are actually substantially smaller than that, even though they all have percent signal changes of, above 1%, which are big for fMRI. So what this is telling you is that the average study that you see in the literature last year was only or the median at least, was only well enough powered to find the biggest effect you would ever expect to see in an fMRI study. Most realistic, interesting effects are probably an order of magnitude smaller. So we definitely have a power problem. And if we actually look at, this is plotting the sample sizes of the studies that were part of that Mueller meta-analysis. And the ones in red I'm plotting here I'm calling them grossly insufficient N, based on this argument by Simmons et al. In their false positive psychology paper that nobody would ever plan to collect less than 20 observations per cell, unless it costs so much, to collect those data that you actually justify it. And in general, if people are reporting sample sizes smaller than 20 per cell, they're usually probably doing some kind of interim flexible termination rule where they're testing, doing statistical tests after every subject or after every few, and then stopping when they get a significant result. They showed in that paper that those types of flexible termination rules lead to highly inflated false positive rates. Now, we also know now that when you have small samples, you can get highly variable associations. And this is, one example is this Merrick et al. Paper that came out a couple of years ago that just showed, actually, they didn't, we knew this from statistical theory a long time before, but they just showed with real data that when you have small samples, even if the true association, which we now know that true brain behavior associations are probably, most of them are very small, at least when it comes to connectivity or structural differences. But with small samples, you can get huge variability in the estimated effect sizes. And that if you have any kind of publication bias, then you can basically get highly inflated effect sizes in the literature. Okay, so I hope I've convinced you that we need big data to help us get around these reproducibility problems. Now I want to convince you that we need it to empower the kind of AI tools that we think are going to be really useful. So in the last couple of years, people in science have become very excited about taking the kind of models that we all now use every day, right? What have come to be called foundation models. These are models like GPT-4, Claude, DALL-E that are trained on large amounts of data, like internet scale data, usually trained in some sort of self-supervised way. So you don't actually need training data. Other than just the data themselves, you don't need any kind of labels. You can just train it, for example, to predict what's the next word in the text. And once they're trained, they can be adapted to all different types of problems, right? You can go now to GPT-4 and ask it to do all sorts of things that it was never trained to do. It was just mostly trained to predict the next word and then to kind of be nice. And these types of models require massive data, right? So this is a plot over time of large language models showing on the y-axis how many parameters are in the model. So you see we have models now with over hundreds of billions of parameters. And then the number of tokens, which are roughly words, that they're trained on. And you can see now they're in the order of trillions of tokens, right? So to get models that are as performant as the large language models that we all work with, you really need, an astounding amount of data. And we know this from a lot of work on scaling laws. We a couple of years ago started trying to build these sort of foundation models. This is work from Armin Thomas in my lab, published at NeurIPS a couple of years ago, where I'm going to talk about Open Neuro in a bit. We took a relatively, what sounds like a lot of data, like about 12,000 fMRI runs, 1,700 subjects, but really a tiny amount of data compared to like LLM scale. But we basically took, various types of architectures like those that are used in language models and ask, can we build a model where we train it just to predict the next time point in an fMRI time series, but then use that model once it's trained to decode what kind of task a person was doing? And what we show is that, and I'm happy to say more about this in the discussion. I'm gonna go through it quickly. But basically, compared to the kinds of decoding models that we would have used beforehand, these models, especially the one that is a GPT type model, can do much better decoding from very small data sets compared to those linear models by leveraging the representations of the data that model learned. And we're really excited about pushing this sort of stuff forward. Now, fortunately, the amount of data that we have in the neuroimaging field has really exploded. Right now, there's now a number of large studies that share data, very openly, like ABCD, Human Connectome Project, Indy. I'll talk about Open Neuro. Biobank doesn't share it quite as openly, and they've actually kind of clamped down on what you can do with their data. But unfortunately, we know that there's tens of thousands of subjects of data out in the world that we can take advantage of. Unfortunately, none of these focus on the collection of phenotypes that are relevant to women's brain health. So I'll show you an analysis of the Open Neuro database. So this is a database we started back in 2017. It went live. Now has over 55,000 subjects worth of data from over 1,200 data sets, mostly MRI, but a number of other data types as well. And so we're now over, we've been growing kind of, really steadily over the last 8 years now. But again, we can look for those same terms that I showed you before and see how many data sets actually show up that match a search for those terms. So again, if we search for, bilingualism, music, and mental imagery, music is actually really prevalent. And that might be people are saying, well, we played music for the subjects during the scan or something like that. They're not necessarily all studying music, but nonetheless, they're just in terms of what they mention, you get a decent number of data sets. Again, the number of data sets matching these women's brain health relevant terms are just, way below what you would expect, given the number of people who are impacted by these things compared to the number of people who are impacted by these things. So the goal of the WBHI is really to address this by generating a large data set that's enhanced with relevant phenotypes for women's brain health. So the goal is really to kind of capture what you might imagine is kind of like the scientific exhaust of a bunch of imaging centers, right? Basically pulling in people who show up for any old brain imaging study about whatever. They're all going to be getting a structural MRI, T1 weighted structural MRI. Many of them are going to be getting a resting functional MRI scan. They might also be getting other types of scans. So the goal is to, and I'll say more about how this works, is to basically recruit subjects from all these sites, have them give us a little bit of extra phenotypic data, and then share those data with the world. So, there's really three foci here. One is the data. Another is the infrastructure, and that's my group's role in the project primarily. I'll say more about that. And then the AI tools that will take advantage of the data that's generated through the infrastructure. So, okay, so I want to make the point that sharing data is great, but in order to make the shared data useful, you have to work on infrastructure. Infrastructure is I refer to it as plumbing. It's the kind of stuff that my lab's been doing over the last 10 years. Often feels like digging through pipes to unclog them. But it's turned out to be incredibly important, I think, for making data sharing effective. And a lot of it is based around a set of ideas that have emerged in the last 10 years called the FAIR principle. So FAIR stands for findable, accessible, interoperable, and reusable. So the idea is like, if I'm going to put data out, one, people need to be able to find it, right? It needs to be linked in a way that the world can find it. Two, the data need to be accessible. They don't necessarily have to be fully openly accessible, but I need to at least know how I can get to the data or who I need to ask for permission to get to the data. They need to be interoperable. If I give you a file that's a bunch of hexadecimal code with no information about it, that's not useful for you. Or if it's in a file format that the software doesn't exist for anymore, that's not very useful for you. And finally, it needs to be reusable. And what this really refers to is you need to know what your rights are. What's the license under which you're receiving the data, so you know what you are legally allowed or disallowed, or what the expectations are for you in using the data. So we've spent, actually the first meeting to develop BIDS happened just over 10 years ago, January 2015. So we've spent the last decade trying to build this thing that's like this this beautiful piece of plumbing called the brain imaging data structure. So what BIDS is basically a standard for organizing files and metadata in a neuroimaging data set. So when data come off the scanner, they look like this. They're in a bunch of like, kind of indecipherable DICOM files. What BIDS does is it provides you guidelines for saying, how should you organize your folders? How should you name your files? And one of the really important parts of that is that it makes this data set be machine readable. So now I don't have to know anything about the data set. I don't have to know anything other than what's in the data set. If you give me a properly formatted BIDS data set, I can point my software at it and it can load it and work with it without a conversation. So one of the great things about BIDS was that, Chris Gorgolewski, who was really the initial driver of it, had this idea early on that we should make it extensible. And the community has really jumped in and done all sorts of work around extending it to things like MEG, EEG, intracranial EEG, quantitative MRI, PET, FNIRS, ASL, all these different things. It's work of a ton of people. This is a paper that we just published that kind of overviews it, and I would point you to this in the New Imaging Neuroscience Journal. But I just wanted to highlight how many people have played a role. We basically opened up authorship to anybody who's contributed. And so we have over 100 people who've played a role in the project. So a ton of work by a ton of people. But I think it's really made the ability to share data really powerful. So if you go to Open Neuro and you log in and you hit this upload button, the first thing it's going to do is say, select a BIDS data set. And when you click that button and select a folder, before it ever uploads anything, it's going to look at the data set. We have an automated validator that can look at a data set on your machine and say, is it a valid BIDS data set? And if it is, you can click upload, and within a few minutes, you can have it publicly available either to your collaborators or to the world, with a DOI that you can put in your paper to link to it. So let me walk you through a little bit about the details of the workflow that we plan to use within the WBHI. And we call this the WBHI base study. This is the one that is going to be collecting this kind of neuroscientific exhaust from lots of different sites. We have a number of people who are, across UCSB and Stanford and all the other sites who are playing a big role in this. And then we have a set of tools, and I'll talk a bit about what those tools do. So here's what the workflow looks like. So a subject shows up at any of these sites and is, consented to do their study. And that brain imaging center gives them, a number, a study-specific number, and then they collect their data. After the scan, the subject is approached about, participating in the WBHI. And if they're interested, they go to a survey run on REDCap, which is a secure platform for, for kind of data management. And they consent and they complete a survey. And then what we do is using a system called Flywheel, which basically is attached to each of the imaging centers. Basically, those data are de-identified and pushed to the Flywheel database. And then we basically pair up the Flywheel data with the data in REDCap, which live at Stanford, and then do a bit of processing on those data. And then they're basically pushed to Open Neuro. And then over time, we will, release those data in an open access way. We'll, some of the phenotypic data that would be considered more sensitive will be released under more controlled circumstances. The imaging data will be released, and some of the phenotypic data will be released open access, fully open access, with no requirements other than agreeing not to try to re-identify the data. So the base study will collect basically anybody who shows up, all sexes, all genders, any adult, and basically the idea is to collect data from typical controls. And really this means that anybody who doesn't currently have a diagnosis of a neuropsychiatric disorder. They go home, complete the REDCap survey online. We've tried to build this thing to make it as easy for subjects as possible. So, the skip logic allows that they only have to see the relevant questions. Takes anywhere from 5 to 15 minutes, depending on, how you answer things. And they get a $10 gift card via email once they complete that. So Flywheel, as I mentioned, is a cloud-based, commercial cloud-based data management system, which we've chose to rely on because they have really seamless tools for grabbing data from a scanner and pulling them into the database that lives in the cloud. And they can do this in a way that can meet the security requirements of these brain imaging sites, which has been the biggest challenge of implementing these, is just like dealing with the different security requirements the different imaging sites have. And then they can, we have kind of granular permission on Flywheel. And so then what we do is grab any non-task data. We're not collecting task fMRI data in part because the metadata requirements for that is much higher than for things like structural or resting fMRI. We pull that into our WBHI project within Flywheel, and then those get converted to the BIDS standard and pre-processed. Then basically those data get paired up with the OpenNeuro database, and we're going to be releasing those in waves. The first release will be coming at some point later in this year, hopefully by Q2. So one of the really nice things about BIDS is that it allows us to build tools around it. And we've developed now a really nice ecosystem of open source tools that can allow us to do a lot of the stuff that we need to do with neuroimaging data in a really robust way. And one of the first things we have to do is, assessing image quality. On the left here, you see is like a really striking example of a kid that's moving around in the scanner during an fMRI scan. And you can see just how, badly, I mean, this data set would not be salvageable, but there's a lot of data sets that have, a degree of motion that's not this bad that can be corrected and salvaged. And for structural imaging as well, there's lots of different artifacts that can occur in structural imaging related to head motion and various other sorts of things. So we have this set of tools we call BIDS apps, because they're basically software applications. You can point at a BIDS data set, and you don't need to tell it anything else. It uses all the BIDS information to automatically process the data set. So one of the tools we developed, and this was developed by Oscar Esteban, who's now in Lausanne, Switzerland, is called MRI-QC, which provides both group and individual reports on a bunch of different quality control measures. So you can look at the distribution across subjects, and let's say you see, well, I want to know what's going on with one of these outlier subjects. You can click on their data point, and then you'll see, their individual images with a lot of additional information. For fMRI, we have a lot of, mosaic plots and things like that can allow you to pretty easily dig in on what might be going on that can cause quality assurance problems. And so we think this is really important given the potential for problematic images to derail analysis. Another tool that we developed is something called fMRI Prep, which is meant to be a highly robust tool for the pre-processing of, it's primarily meant for fMRI. It also does a lot of structural imaging processing, leveraging tools like FreeSurfer. We also leverage. There's a lot of open source tools in the imaging world that we leverage for this, like FSL, like AFNI, like ANTS. So this is really kind of a workflow that wraps a bunch of existing tools. And we're constantly testing this on lots of new data sets because new data sets can break it. And in fact, we have a telemetry system that tells us, people can opt out of it, but if you don't opt out, it'll tell us when you run fMRI Prep, basically, did it work? What kind of data set did you try to run it on? Did it work? And if it didn't work, what was the error that caused it to break? And you can see, now there's well over 1 million data sets that have been processed. And consistently over the last, more than a year, we've been at like around 8,000 successful fMRI Prep runs per week. It's successful about 2/3 of the time, so it crashes about 1/3 of the time. And some of that might be just people killing it because it's taking too long. We haven't done a deep dive on why it crashes, but we do look for, when we put out a new version, we look for new errors, and it allows us to kind of, keep track of what might be breaking. I want to last just sort of mention some of the special challenges of multi-site data for statistical modeling and data analysis, and highlight some of the work that our group is doing. Lately it's become a big focus of our group. And particularly Eric Bridgeford, who's a biostatistician, who joined the group last year, has been doing a bunch of work on one on understanding demographic site differences. So these are data. This is from the ABCD study, which is a multi-site study across what 21 sites, I think, um, just showing for a bunch of different, demographic or phenotypic measures, the distribution. Each of these dots is a site, and you can see that, for some of them, actually for gender, it's pretty a small distribution, but for some things like the racial variables, parental income, like SES, even interesting things like were you exposed to anesthesia as a child? There's lots of, and even the age within the small age range. There's lots of differences across sites, and we know from a lot of work starting in genomics, decades ago, that these types of site differences can have big impacts on analyses that combine data across sites because any sort of any like if you're collecting data on different MRI scanners, even though, well, actually in ABCD, they even have different, makes and models of scanners. And then combining, but even if it's the same make and model, we know that two different Siemens Prismas can give you slightly different data. And so those differences, if they're correlated with demographic differences, can then kind of propagate throughout your analyses. Eric published, this is from his dissertation, published this. No, actually, sorry. This is from a paper that we have submitted that is meant to lay out this kind of like global view of how to think about processing data in the context of side effects, particularly from a causal inference perspective. But here the point is that there's a lot of stuff we know about particular covariates and phenotypes that are going to be related in our imaging measurements, and then we can think about taking those into account when we actually process the data. And trying to fix these kind of batch effects. And he has a paper that just came out from his dissertation work in imaging neuroscience showing that some of the most commonly used existing methods like combat are actually can be problematic under certain types of distributions of confounds. And so he demonstrates that there's a set of methods using causal inference tools that can try to address that. Another really big problem is that there could be confounders that we haven't measured, right? There's often going to be things that we just don't know might be important. And so within the field of causal inference, there's emergence of a set of tools that can tell you, one, how sensitive are your analyses to particular types of modeling choices, but also how big would an unobserved confounder have to be to explain away your results? So if you see an association, how big would an unexplained confounder need to be to tell you that association is not real? And then you can kind of use what you know to decide, does that cause us to question the reality of this particular association? Given that, all of the analyses that we're going to be doing with WBHI, just as in ABCD and other large data sets, are going to be association analysis, right? We're not doing causal experiments. We're just looking at the associations that occur. So unmeasured confounding is a huge potential challenge here. Another really interesting question, and this is work by Jocelyn Ricard, who's a grad student in my lab with Keith Humphreys here at Stanford, is the degree to which the ways that we process data can result in selection biases that could end up biasing our data in kind of problematic ways. And she's focused on the degree to which head motion exclusions are racially biased. And it turns out that they're heavily racially biased, right? That Black kids are basically twice as likely to be excluded on the basis of head motion from fMRI analyses compared to white kids, with lesser inflation for kids from other non-white groups. And so we think that this is a big problem because it's causing these structured selection biases in the data, which again, we know that selection biases can have big impacts on subsequent associational analyses. And so these are a set of questions that we're going to be focusing on a lot once we get the data to try to understand, what are the ways in which we can try to correct for these side effects and potential demographic differences, which are almost certainly going to exist across the different sites in the UC. Okay, so finally, I just want to talk about kind of where we want to go from here. And the first place is basically, to the world, right? we have a small number of sites contributing right now. We're trying to loop in additional UC sites, but there are sites around the world that we could, in principle, recruit to be part of this, and we want to try to do that because the amount of data that we could collect would just be exponentially bigger. Second is moving towards refining the phenotypes that we collect. So right now, we basically take all comers in terms of data, right? So every different investigator often has their favorite T1 weighted scan, their favorite diffusion scan, their favorite resting fMRI acquisition. And the fact that all these data are collected with different types of scans is yet another kind of site effect or sub-site effect that we have to think about trying to address, because the different scans are going to have different artifact characteristics and so on. So the goal is to offer at least a common acquisition protocol for groups, an optimized acquisition protocol across sites. And this is made easy by the fact that so far all the WBHI sites within the UC system have the same make and model of scanner. So in principle, they could at least use the same pulse sequences. The other thing we want to think about doing is, we currently have a women's health battery of questions, but there's almost certainly more things that we could think about adding, within the constraints of subject tolerance. We don't want to make the thing take hours, but nonetheless, we're thinking about improving that. We also want to think about adding phenotypic breadth. And so, right now we're focusing primarily on MRI imaging and self-report measures, but we want to move in other directions. And the first step is an ongoing project that was just funded by the Chan Zuckerberg Initiative to start collecting blood data from participants and using that for proteomics and hormone analyses. And this is leveraging this really cool new tool called the Tasso, which is we refer to as the sucker. It's basically a thing you put on a person's arm, and it uses this capillary measurement technique to basically pull out enough blood to do analyses on without actually having to inject them with anything. And then that just gets sent back to the center here at Stanford that processes it, and then will be sent along to various centers for proteomics or hormone analysis. So we're really excited about the potential for having, biosamples alongside the imaging and behavioral samples. We also are thinking about kind of targeted sub-studies. So, within the big study, we could imagine that there would be subsamples where we might say, let's collect a particular type of data, like neuromelanin scanning or MR spectroscopy for quantification of particular types of neurotransmitters. We might want to do kind of extended self-report to find out things like trauma exposure or social network structure. And then we might want to do high density digital phenotyping to get at real world behavior, to look at actigraphy, EMA analyses of mood or cognition, cycle staging, all these types of things that we can imagine doing in a more high density sort of way. So I just want to end by thanking all the folks who played a role in this. Obviously, thanking Emily for her leadership and kind of making this all happen, but then all the other leaders of the project and all the people at all these different sites who are kind of really going out of their way to help make this thing happen. I particularly want to shout out to Josh Buchholz, who's the scientific director and has really, from our side, kind of made the Data Coordinating Center happen. And finally, thank all of our collaborators who helped build all the tools that I've talked about over the last decade, really, and the folks from my group. And I am happy to take questions now.
Thank you very much, Russ, for this very eyes-opening and thought-provoking talk. Very inspiring. We have time for questions. Are there questions in the audience? Well, yes. I'm going to read it. Thank you, Russ. That was great. What opportunities or initiatives within WBHI could enable early career researchers based outside of the US, like myself, to play a more active role in advancing the organization's goals, even if we don't have institutional influence?
That's a great question. So I think there's a lot of stuff that that anybody could jump in and sort of play a role in. And for example, once we start putting the data out, then those data will be available to people around the world, under a very minimally restrictive use agreement. And so you can jump in and start analyzing those data, thinking about what are the new types of analyses, new types of questions. That one might come to ask. You can also think about getting involved if you're from the engineering side, getting involved in developing some of the tools, right? There's always work to be done on projects like MRI-QC or fMRI prep. I think that's going to also be the case probably on some of the AI projects. And so all of these open source projects are open to contributions from people around the world. And then also to the degree that you can start thinking about depending on where you are, and what the local regulations are, it'll be more or less easy to share your data. For people in Europe, there's ongoing work trying to develop like basically a European version of Open Neuro that complies with GDPR, which obviously will require additional steps to get access to data, but nonetheless trying to make those European data searchable so that even if you don't have access to the data, you can search and find data sets that you can then ask for access to and go through all the GDPR hoops to get those data. I think there are a lot of ways that that people can get involved.
Yeah, if I may add also, if you're interested in the algorithms or the machine learning methods, we have partnered with WITS, Women in Data Science, Kaggle, and the Child Minds Institute to launch an international challenge. So we've put one data set really easily findable online, and then you can motivate yourself or your collaborators or your students to participate in this challenge. So that will be a very easy entry point to the type of questions we are interested in.
And we'll have a panel discussion next month that's sort of related to that a bit. So Russ, thank you so much for all you do, first of all, because I don't like collecting data. So thank you, just personally. Susan knows this, and Jared. But I have a question actually specifically about foundation models. So in my mind, somehow foundation models may be used in a sort of a denoising context, and it allows sort of us to boost the SNR, if you will, of fMRI data. So I don't know if have you done sort of simulation analyses of the power, like the amount of percent bold change you need to detect with and without a foundation model to maybe see if there's a boost there? And if not, let's do a paper.
Yeah, no, we have not. I think that's a really important question. I have a new postdoc coming from Helen Zhao's group, joining my lab later this year.
Great, yeah.
And she's going to be pushing on neuroimaging foundation models. So I think she'd be a great person to kind of to help with that. So yeah, let's definitely have that conversation. Because I agree, it's a really important question. We have not asked that at all.
And I think it's especially relevant for clinical MRI, where it might not be the optimal resolution or SNR, right? So you could take these sort of, we've seen in some of our work that we have these sort of really fast acquired fMRI data. It's like 5 minutes of data, which usually you can't get much out of. But if you put it through some of these sort of foundation models or like foundation models, you can get a better signal, I think. So anyway, that might be one of the ways that AI could potentially help with these clinically acquired images.
Definitely.
Awesome. Yeah, very important questions. We have two more questions from the audience. The first one from Summer Meckenkoch said, sorry, such a specific question. Do you like the Tassos? I started using them and then went back to finger poke DBS because of cost and ease of processing.
I can't really answer that question in part because, we haven't started using them at scale. And also Josh Buckholz, who I don't think is on the webinar today, is really the expert in our lab and who could speak more to that. I think the challenge for us is that these, if we're a single site, then I think, doing phlebotomy would probably make sense, or, whatever kind of other collection method. But given the kind of broad scale of the different locations where this is going to be done, I think that it's probably not feasible to do it using a blood draw. But we'll see. We'll ask us, in a year how well the Tasso has worked, and we'll know a lot more.
Thank you.
Yeah, this is a pilot project to exactly answer that question. Is this feasible? Is it scalable? what are the pressure points and can we overcome them working with Tasso and working with the processing end at Stanford. So stay tuned.
Thank you. Next question from Joanne Kenny. I am setting up a research project and want to include a sex and gender focus from an early stage. Are there opportunities to collaborate with WBHI?
Definitely, yeah. And I think, Emily is probably the best person to speak to this. The one thing I'll say is that, the one thing to think about upfront is making sure that when you get when you design the study, that you're kind of harmonizing your phenotypes as well as possible, particularly with the questionnaire that we're using, so that we have common data elements that we can focus on. The other is making sure that your subjects are consented to allow the kind of broad data sharing that we do. And there's a project called Open Brain Consent that you can have a look at that has kind of, template language, both for, US and European studies. So to basically make sure that your subjects have been consented in a way that they understand how the data are going to be shared and what the potential risks are of that. Because I think that's becoming, increasingly important. I don't know, Emily, do you want to say any more?
I'll just add a couple more. So from a sort of training perspective, Amy and Jaren and others are thinking about building an educational workshop that we'll host here on WBHI. So sort of using this space to really kind of walk through best practices and potential pitfalls to doing this. And maybe we'll take specific examples. So if you're in the maternal brain space, or if you're trying to study menopause and you're using UK Biobank data, but really sort of just, yes, think of it like an educational workshop. So stay tuned for that. We're putting it together, but that will come, and I think would be a great kind of crash course for how to get into it. And then the other thing that Russ mentioned is we are building, we have the WBHI sort of phenotypic battery, but in parallel with that, Winnie Orchard, who's Director of Scientific Programming for the WBHI, and Laura Pritchett, and a whole team behind them are working on, and many of whom I see sort of on this call, we have a whole incredible group of scientific advisors, are trying to build what we hope will be the gold standard kind of metadata to collect to capture kind of lifespan phenotypes relevant to women's brain health. So that is a labor of love that is ongoing, and once it's established and it's vetted by the scientific community and it's published, it's going to be open access, and we hope again it will be adopted broadly so that everybody's collecting the same kind of data in a way that's harmonizable.
Awesome, thank you. I don't see any question in the audience right now. Oh, Sarah?
Yeah, I have one question about the Open Neuro. It was an amazing talk. Thank you so much. When you were mentioning about Open Neuro, you were talking about there were typically controls. Are you planning to include people with other neurological disorders like Alzheimer's disease and MS, which are more likely to be seen in women?
I think that's an interesting open question. I, in theory, don't see any reason why we couldn't do that. But we're kind of starting with a more limited sample. But I think ultimately, given that those are, important, populations for women's brain health, it seems like we should consider that. And then the question is going to be, do we want to do that in a targeted way or in a broad way, just kind of taking everybody? I think that my guess is that, in order to get large enough samples of things like MS, you really need a targeted acquisition, right? Because most studies at these imaging centers are going to be excluding people with neuropsychiatric disorders. And so I think that, we probably need to think about kind of targeted acquisitions to try to address the, particular disorders of interest.
Wonderful. Thank you.
Other questions? I have one actually on the foundation models. Maybe that's something that your future postdoc is going to work on. But do you know of any multimodal foundation models for neuroimaging or neuroscience? So for example, fMRI, but also structural MRI predicting not the next time step, but maybe a different patch in the image, and even adding some, other type of text data.
I should say I don't follow the conferences as closely as I should. So a lot of this stuff might be showing up, and Amy may know more about this than I do. Yeah.
So actually, we have one, and it
Yeah.
hopefully will be out in Nature Methods in the next couple of months. But it's based around the connectome. So it's using both functional and structural connectivity matrices, pushing them into a common latent space, and then kind of using the latent space information to map back out to each modality and also to different demographics and things like that. So
The Kraken.
the Kraken coder,
The
yeah.
Kraken coder. Yeah, yeah,
I
yeah.
like to think of it as a foundation model, but it's not exactly the same as predicting the next time point.
Mhm.
But it's kind of using multiple modalities and pushing them together to a common latent space and preserving inter-individual variability. Yep.
Yeah, that's fantastic. Thank you. I have another question on AI and reproducible science. So you showed this analysis of 99 paper from fMRI data that were finding completely different effects. Are they plans, or do you know of any analysis that does that on AI papers? Because there is a lot of papers that are published on AI and ML every day. By month, I think it's over 4,000 a month that have the keywords AI or ML and put on archive.
Yeah.
Should we
I mean,
try
the thing that comes to mind foremost is the work by Sayesh Kapoor and Arvind Narayanan that's been looking at the potential. I mean, I think by far the biggest, I don't know if you agree with me, by far the biggest problem in the application of machine learning and AI tools is, leakage of test set information into the training set. And they show, and there's a lot of examples of this, right? But they, in their paper that was in Patterns, I think a year or two ago, show, across a lot of different literatures, evidence of this kind of leakage. And then there's a lot of individual meta-analyses focused on particular domains that show evidence of leakage. And we've certainly seen, very prominent examples in the imaging field, papers retracted that made splashy claims on the basis of what turned out to be, kind of overfitting. So, and then, the other piece of work that comes to mind is actually from a number of years ago. I don't know if it was ever published, but Joël Pineau had a talk at NeurIPS back in like 2019 that you can find online, right? Which went through all these examples. This was focusing on like reinforcement learning, but basically showing like, you know, for the same for the same type of problem, you can see three different methods performing very differently. So for the same method, you can see three different implementations performing very differently. For the same implementation, you can see performance differing greatly depending on the random seed. And I think so. Random seeds are another place that I worry that there's a lot of like kind of seed hacking, people trying, and maybe this is a benefit of these models that are so large that you can't run them 20 times and find the best performing one. But nonetheless, I think that kind of random seed hacking is a huge problem as well. Then I don't know of papers that have looked at that. I mean, the other thing that comes to mind is simply, the kind of overfitting to large data sets that seem to be happening. I don't know if you know this work by Ben Recht and Ludwig Schmidt, where they went out and basically generated new versions of ImageNet and CIFAR-10 using basically, they said, we're going to use the same rules that they use to get images, but they go get a bunch of new images, and
That's
all
awesome.
the models perform much more poorly on these new data sets than they do on ImageNet and CIFAR-10. So clearly there's a lot of overfitting even to these very large data sets.
Yeah, and the model are so expensive to train in terms of time or just CPU resources. And if we were to want to try to reproduce everything, that would cost so much money that it's almost impossible to do that. So big problem. Okay, there is one last question from the audience. Are there any plans to make the preprocessed data available that could be helpful for people new to neuroimaging? Also, will the genomics data be made available too?
Okay, so on the imaging side, yes, definitely. In fact, we right now are going through Open Neuro and pre-processing using MRI-Q-C and fMRI prep and releasing those pre-processed data for as many data sets as we can. We've done over 200, I think, at this point. We've got another 100 in the hopper. So we will have, a large amount of pre-processed data, and the WHI data will definitely be, we will be releasing pre-processed versions of them. I don't know if we'll be able to do it with the initial release, but definitely soon thereafter. So we're not actually collecting genomics data yet. We're going to be doing proteomics and hormone analyses, and those will be shared maybe under a more restrictive license, depending on the guidance that we get from our legal team. But they definitely will be shared under some kind of agreement. And so, yeah, so that will definitely be open. We may include genomics later. The problem with genomics is you really need very big samples to do anything useful. But if we could team with other groups that are already collecting genomic data, there might be some really interesting things to be done there.
Awesome, thank you. Well, it's 9 my time. That concludes the seminar for today. Sharon, do you have any announcement for next time?
Yeah, we will have a roundtable on computational neuroimaging in women's brain health, and we will have Dr. Katie Chang, Amy Kuzelski, Nina Milon, and Russ Poldrack, and Monica Rosenberg, and Ted Satterwait. I don't know. Sorry if I mispronounced. So we will have a roundtable to better understand the best practices when collecting and analyzing women's health neuroimaging and emerging technologies and analytic tools, specifically for women's brain health studies. So it is on March 10th next month.
Awesome, thank you. Yeah,
How
join
much,
us
Russ?
on March
That was
10th.
brilliant.
Thank
All right,
you.
bye, everybody.
Bye.
That's great talk. Thanks.